Good morning, everyone. The topic of my presentation today is the neuroepigenome, implications of chemical and physical modifications of the genomic DNA in schizophrenia. So first of all, what is schizophrenia? Schizophrenia is a severe mental illness that affects around 1% of the population in the world. Now, schizophrenia is a highly heritable disease, around 80%. A clinical diagnosis of schizophrenia is based on the qualitative terms defined in the DSM-5 as some positive symptoms like hallucinations and delusions, negative symptoms, avolition and withdrawal, and cognitive dysfunction, deficits in executive function. Now, because of a limited understanding of the molecular basis of schizophrenia, we do not have any biomarkers to facilitate the diagnosis or to monitor disease progression. Now, what causes schizophrenia? What is the etiology of schizophrenia? Now, it can be nature, that is genetics, or nurture or environment, epigenetics, or it can be neurodevelopmental or neurodegenerative. Now, the answer to this, the cause can be partly all of these. Now, this is the old, slide, it shows the old theory of mental illness, which has been long abandoned by the modern theories now. Now, according to this theory, that there was a single abnormal gene that would be inherited, and that would lead to the production of an abnormal gene product, and that would lead to neuronal malfunction, leading to mental illness. Now, this theory, it is shown by a big red cross sign, indicating that this is now not at all used. This theory is not accepted now. So according to the modern theories, the current theories, they suggest that people, they inherit the risk to a mental illness, but they do not inherit the mental illness per se. That means, for example, if we inherit an abnormal gene, but that means we will inherit the risk of developing that mental illness, but not the mental illness per se. And whether that mental illness will occur or not, it will depend on what happens to us in our environment. For example, substance abuse. Now, this is the modern theory of schizophrenia, of any mental illness, here schizophrenia. So we have nature and we have nurture. Nature is genetics. So we have thousands of genes in our body, to be very precise, 20,000 in our body. So out of those thousands of genes, a few hundreds of genes, they regulate the neurotransmitter system, the synaptogenesis, neuroplasticity, cognition, neurotoxicity of psychosis, and stress vulnerability. They together constitute the polygenic risk score. But now this is not only, this confers only a part of the etiology of schizophrenia. Now, what is the other part? The other part is nurture, or the epigenetics or environment. So whenever we'll inherit any abnormal gene, but that would not, we'll inherit only the risk of developing that mental illness. Now, whether that mental illness will manifest into a illness itself will depend on this, the nurture. So we can have any obstetric events, abusive childhood, virus, marijuana, traumatic experiences, and bullying. Now, together, these both will lead to the will lead to the causation of a mental illness. Now, how does environment unmask schizophrenia in those who have genetic risk for it? The answer to this is epigenetics. Now, here we'll be discussing very briefly about epigenetics. So, epigenetics, it is basically the study of how our behavior and how our environment, it affects the way our genes work. Now, epigenetics is different from genetics. Epigenetics is above genetics. In genetics, there are changes in DNA sequences, but in epigenetics, there are reversible changes. They do not change the DNA sequence, but it determines whether a gene will be expressed or not, how our body will read a gene. Now, these they survive cell division. That means epigenetic changes can be passed from generations to generations, but they're still reversible. They are not irreversible. Now, for example, now we have around 20,000 genes in our body. Now, all those 20,000 genes in our body, they are not expressed. Why? Because even though they are inherited, but they are not expressed, the answer to this is epigenetics. Now, epigenetics determines whether any given gene is actually made into a specific RNA and protein, or if it is silenced and ignored. So, the story of our brain depends not only upon which genes are inherited, but also upon the abnormal genes are expressed or not. Even normal genes are silenced when they should have been expressed, or they become expressed when they should have been silenced. So now we know that why identical twins developing from the identical twins or monozygotic twins having 100% of the same genes, having 100% same DNA, why do they have different personalities? The reason to this is epigenetics. Now, before going into the molecular mechanisms of epigenetics, we'll be very briefly discussing about genome. So genome is a complete set of genes or genetic material present in the cell. In this diagram, for example, we can see we have 23 pair of chromosomes, including the X and Y, which are present. Basically, they are condensed into the chromatin, and the chromosomes are present in the nucleus of the cell. Now, in this diagram, here we can see this is a double-stranded DNA helix. Now, this double-stranded DNA helix is wound around the histone proteins. The purple ones here, these are histone proteins. Basically, these histone proteins, they are present in octamers, means 1 unit constitutes of 8 parts. Now, these histone proteins, they are basically the structural, and they provide structural support to the DNA. Now, DNA is wound around these histone proteins. Now, this single unit is known as a nucleosome. Now, a lot of nucleosomes, multiple nucleosomes, when they condense together, they form the chromatin. This is the chromatin. Now, since the chromatin is very large, it is very big, so it has to be condensed. It has to be physically compacted so as to be incorporated into the nucleus. Now, this chromatin, this can wind and condense into the chromosome, or it can even unwind when replication or transcription needs to occur. Now, changes or modifications in this chromatin leads to the expression of a gene or silencing of a gene. Now, this we've already discussed that inside the nucleus, around 147 base pairs of DNA are sequentially wrapped around octamers of histone proteins called as nucleosomes that fold in a hierarchical manner into individual chromosomes. The nucleosomes that mediate compaction of DNA are condensed into the chromatin fibers that fold into individual chromosomes and occupy distinct regions within the nucleus called as chromosome territories. Now, epigenetic control over whether a gene is expressed or silenced is done by modifying the structure of chromatin. Now, here we can see this is the octamer, the histone protein, and the DNA is wrapped around it. Now we come to what are the molecular mechanisms of epigenetics. How is a gene expressed and how is a gene silenced? That basically depends upon modifications of the DNA and histone. Now, chemical modifications include methylation, acetylation, phosphorylation, ubiquitination, and sumoylation. Out of all these, methylation is the main one. So when DNA and histone, they are methylated. So what basically happens is when chromatin is present loosely, so it is not physically compacted. So that is, it is available for transcription purposes. So that chromatin is known as accessible chromatin. Now, if that chromatin, including the histone and the DNA, when it is methylated, methylation leads to the compaction of chromatin. Now, when a chromatin is compacted or physically condensed, it will not be available for molecular transcription, and that is known as inaccessible chromatin. So methylation is the main process which leads to either expression of a gene or silencing of a gene. Silenced DNA means molecular features that are not part of a given cell's personality. Now in this diagram, we can see this is the accessible chromatin, as I said, the open chromatin, which is available for transcription and would lead to gene expression. And this is the condensed chromatin. The pink dots here, they represent methylation. Now methylation has caused compaction of the chromatin, and this will not be available for transcription purposes and would lead to gene silencing. Now in this diagram, first of all, let us see gene silencing. So here, these are methyl groups in the color orange, which are present on the histone and DNA. So we have certain enzymes like DNMT, DNA methyl transferases, or histone methyl transferases, which lead to methylation. And since we discussed earlier that methylation would lead to silencing of the gene and no gene product will be formed. Now, similarly, if we have DNA demethylase or histone demethylase, so this will lead to, there will be no methylation and a gene product will be formed. Now, where is this methyl coming from? This methyl is coming from L-methylfolate, which is further coming from S-adenosylmethionine. Now also we have another enzyme here, histone deacetylase. So basically it is said that deacetylation of histone leads to methylation of DNA, which would further lead to silencing of a gene. Now, large scale, basically a lot of studies have been done, and many studies, in those large scale studies, they have identified the loci of schizophrenia on genes. But epigenetic studies, they have still to be done. They are not so much done. And so also, approximately 90% of the schizophrenia associated risk loci, they are located within the non-coding regions. So basically in DNA, we have coding regions and we have non-coding regions. Coding regions constitute around 1 to 2% of the DNA, and non-coding forms the majority of DNA. Now earlier it was thought that non-coding regions, they are junk DNA. But now it is known that non-coding regions, they basically have introns and exons, and introns are spliced to form the mRNA. They also have promoter regions, enhancers, insulators, telomeres, which have a very important role in DNA transcription. So the core pathogenic features which in schizophrenia which have been identified are altered glutaminergic functions, mainly the glutaminergic neurons, impaired synaptic plasticity, and abnormal brain development. These are the three main core pathogenic features which have been identified by various studies. Now, what is heritability? So just going very briefly, heritability can be defined as the proportion of phenotypic variance which is attributable to a genetic variance, or more easy definition is the extent to which genetic individual differences, they contribute to individual differences in observed behavior. Now, because heritability is a proportion, its numerical value ranges from 0.0 means genes do not contribute at all to the phenotypic individual differences, to 1.0 that genes are the only reason for individual differences. For human behavior, almost all estimates of heritability are in the moderate range of 0.3 to 0.6. Now, what are identical twins and what are fraternal twins? So identical twins, also known as the monozygotic twins means one egg is fertilized by one sperm, one zygote is formed, and that zygote it divides into two embryos. So these identical twins have 100% same DNA, and these twins come in sets of either males or females. Now, whereas fraternal twins or dizygotic twins, two different eggs are fertilized by two sperms, and these are genetically similar to ordinary siblings. They are sharing around 50% of the DNA. Now, what is concordance and discordance? Now, concordance is these two terms are basically used in research for twins. Now, if the twins are said to be concordant when they both have a similar trait or they lack a given trait, and discordance occurs when a similar trait is not shared by the other person, as seen in a dizygotic twin. Now, the concordance rate. Concordance rate is a statistical index that researchers used to determine the relative influence of nature and nurture. It is the percent of cases in which both members of a pair have a particular attribute. So in a study of relative influence of genes and environment on a mental illness, the mental illness is the attribute and pairs studied are usually the twins. Now let us come to the hierarchical organization of epigenome. So here we can, at a megabase level in the nucleus, we have compartment A and we have compartment B. So compartment A has open chromatin, or as we said, accessible chromatin, which is available for transcription purposes, and compartment B has inaccessible or compacted chromatin. Now, at a sub-megabase level, we have these units, which are known as TA. These are topographically associated domains. Now, these topographically associated domains, they are basically isolated from neighboring chromatins by bound by CTCF, a transcription factor which is known to regulate a number of cellular processes, including transcription and chromatin structure. Now, TADs play an important role in gene regulation because studies have shown that deletion or disruption of TAD boundaries promote aberrant enhancer promoter interactions, resulting in misregulated transcription. Now, cell type and tissue-specific atlas of the epigenome. The human brain consists of a heterogeneous population of neuronal cells, which are embedded in a field of functionally distinct glial cells that provide structural and metabolic support to neurons, such as astrocytes, oligodendrocytes, and microglia. From decades of research, we have a broad understanding of the morphology and major classes of neurons, but taxonomy maps are not available for epigenetics. Now, FANS is a process, basically fluorescence activated sorting of fixed nuclei. It is a method for studying nuclei from specific cell populations that preserves post-translational modifications. Here in the subsequent slides, we'll review the research efforts that have created comprehensive maps of brain cells using fans. Now, yes, sir. Yes, sir. So basically, in research, there is a process which is known as FANS, which is fluorescence activated sorting of the nuclei. So this technique has been used to take different cells from different brain regions at different developmental stage in humans. And so basically we isolate the nuclei and then we study that which chromatin is methylated, which cells are methylated, which part, at what developmental stage they are methylated. So this technique will be used for that. So for example, there was a study to investigate the cell type specificity of DNA, the type of cell which has DNA methylation in different regions of the brain. Rizarti et al. Profiled genome-wide methylation in neuronal and non-neuronal nuclei isolated through fans from functionally distinct regions, that is dorsolateral prefrontal cortex, anterior cingulate gyrus, hippocampus, and nucleus accumbens of postmortem brains from healthy control subjects. Now, since he isolated various nuclei from different parts of the brain, it was seen that neuronal nuclei were differentially methylated as compared to non-neuronal nuclei and were associated with heritability of neuropsychiatric traits, including addictive behavior, schizophrenia, neuroticism, suggesting a link between disease and neuron-specific epigenetic regulation in distinct brain regions. There was another study which included the ATAC sequencing, that is assay for transposase accessible chromatin. Basically, transposase is an enzyme which cleaves DNA. So it is a method used to identify open chromatin regions of the genome. When it was applied to fans sorted nuclei of the glutaminergic neurons, GABAergic neurons, oligodendrocytes, and microglia, isolated from 3 different brain regions, it found that accessible chromatin in glutaminergic neurons is enriched with schizophrenia risk variants. So this finding provided additional support to the glutamate hypothesis of schizophrenia, which proposes that morphological alteration of the dendrites of glutaminergic neurons may be present in the cerebral cortex of patients with schizophrenia. Now let us come to neurodevelopment and neuroplasticity and schizophrenia. So basically, first of all, what is normal neurodevelopment? So there is conception, then stem cells differentiate into immature neurons. Now, out of all of these immature neurons, some of the neurons, minority of the neurons are selected for inclusion in the developing brain. Now, what criteria the brain uses for inclusion of these neurons is not known. It is still a mystery. Now, other neurons which are not selected, they naturally die off by apoptosis. Now, it is said that abnormalities in the process of this neuronal selection could be a factor for neurodevelopmental disorders like autism, intellectual disability, ADHD, dyslexia, and schizophrenia. And the selected neurons, they migrate and differentiate into other type of neurons, after which synaptogenesis occurs. Now, most neurogenesis, that is neuron formation, neuron selection, and neuron migration, they occur before birth. And after birth, there is differentiation, myelination, and synaptogenesis, which occurs throughout the lifetime. Now, disruption of this neurodevelopmental process can hypothetically result in various psychiatric symptoms. Now, synapses. Synapses are normally formed at a furious rate between the birth and age 6. Although brain reconstructing occurs throughout the life, but it is most active during late adulthood and adolescence in a process known as competitive elimination. So it is said that this can be a reason that the positive symptoms of schizophrenia, which we see mainly in the late adolescence or just during the early adulthood, that can be due to some impairment in this process in competitive elimination. So this competitive elimination, basically it is a process and it leaves around only half of the synapses that were present during childhood to survive into adulthood.
It is late adolescence and is not in adulthood.
Yes, ma'am. Now, how does our brain decide which synapses to keep and which to eliminate? Normally, when glutamate synapses are active, their NMDA receptors trigger an electrical phenomena which is known as long-term potentiation. Now, long-term potentiation, it leads to structural and functional changes of the synapse that make neurotransmission more efficient, that is strengthening of synapses. Now, strengthening of synapses is said to occur when AMPA receptors for glutamate increase. So, strong synapses with efficient neurotransmission and many AMPA receptors, they survive, and weak synapses with few AMPA receptors are eliminated. Also, the most critical synapses in our brain, which are frequently used by us, they are strengthened and they survive the ongoing selection. And the synapses which we do not use very often, they are not strengthened, and the weakened synapses, they are eliminated. Now in schizophrenia, the pathology is, it is said that the critical synapses are not strengthened. So when the critical synapses are not strengthened, so it is thought that these are weak synapses and these are eliminated by the brain. Now these critical synapses which were important for communication purposes, these are eliminated. So this leads to, it is said that this leads to some pathology of schizophrenia. Now, neurodegeneration and schizophrenia. So basically, it is also said that schizophrenia is a downhill process. So if a patient does not take treatment for schizophrenia, or there is long-term psychosis which is untreated, it leads to neurodegeneration. For example, early there is, first of all, there is prodrome, or the patient is asymptomatic. Then there is a first episode of schizophrenia and the level of functioning, the cognitive decline occurs. But in the first episode, when robust treatment is given to the patient, the effect is, the treatment is very good and the effect of the drugs is very good. But as the patient is non-compliant towards the treatment, the episodes keep on occurring again, and there is progressive brain tissue loss. And after some time, there is treatment resistance. So this is the downhill process of degeneration in schizophrenia. My references are these. Thank you.
Thank you, Dr. Asta, for the presentation. I think we deliberately did this for a recently article which came in Biological Psychiatry. Biological Psychiatry is one of the premier journals. So then we requested that she touch upon the basics of genetics in schizophrenia, and then discuss about making this as an anchor article, and then discussing about neurodevelopmental hypothesis and neurodegenerative hypothesis. I think now we are open for questions, comments, and clarifications from the audience, because all these are very important. MCQs, your exam questions, residents also, because this will come as a long question, a short note. So this is a very important. And genetics, generally genetics is not very easy to comprehend. So I suppose there must be a lot of questions you can ask.
So basically, we have two types of twins: identical twins and fraternal twins. Now, identical twins are monozygotic twins because one sperm, it fertilizes one egg, and there's a zygote, and then it differentiates into two embryos. So both of the embryos will have 100% of same DNA. Okay, but still, what we see is, even though they have 100% same DNA, they develop different personalities. Now that occurs because of epigenetic changes, as we said. Now, what is concordance term is basically used in research purposes for twins. That means when both the members of the pair, means both the twins, either they'll have the same trait, they will have a trait, or they will lack a trait. So this is known as concordance. These are known as concordant twins. If they have the same trait, or they lack, that is concordant twins, and if they not have, that is discordant twins. So concordance is basically seen in monozygotic twins.
Concordance is not necessarily it can be the siblings also if they have the same trait. Typically also they are there. So we actually asked her to put this concordance and discordance, in the sense that concordance is given, there are two individuals who are sharing the same genes and the percentage with which they'll develop the disease. So, and that is concordance. Discordance is they'll not develop the disease. So when you do this and then you minus it, then you get the heritability. Heritability is a very interesting concept. Like heritability of schizophrenia is what? Schizophrenia is like 80%. When we see breast cancer heritability, like BRCA genes and all, and that is around 50%. But still, breast cancer is more genetic than schizophrenia. Schizophrenia heritability is more. That doesn't mean that it is purely we're talking about genetic genes being transmitted and the expression is seen. So there is this epigenetic interactions coming. That is why heritability concept is not purely a genetic concept. So इसलिए ये understanding is needed. So something might be more genetic but can be still less heritable. So एक तो ये है, I think euchromatin and I think heterochromatin. So euchromatin is more available, which is not densely packed. That is what we used to study. Heterochromatin is densely packed, so it is not available for transcription. So there is an modifications in these, and as you said, that they are studying these at various stages by fans. They are trying to isolate it. So this is a new addition, the recent concept. And there was an Professor Sanjeev Jain from NIMHANS. He's the premier faculty in genetics in schizophrenia, bipolar disorders, and severe mental disorders in India. He's a most sought out researcher in this field, and he shared his paper, I think, a few days back. Interesting paper where they actually did the same thing, and they found that neural migration from the stem cells, pluripotent stem cells, the migration is different in bipolar disorders. So they have actually established this in India for the first time. It has been replicated outside. From the Indian context, this has not been studied till date. That is one thing. Methylation obviously is very important part of it. So this also explains that why some people with vulnerability will develop schizophrenia and some people with vulnerability may be asymptomatic like. Any questions? Does drugs have any role in epigenetic modification?
Yes, sir. For example, sir, I forgot to quote an example regarding drugs. So, if it was a study which was done, it was seen that if cannabis is not consumed, so the episode, if it is not the high potency cannabis, it is not consumed, so the risk of first episode psychosis would fall by 50% in Amsterdam and 32% in London. So that means even if they inherit a risk for the mental illness, but if they do not consume the substance, so that basically stays.
Maybe via epigenetic.
Yes, sir.
And then why it is called as neurodevelopmental, as she shared, that in critical phase there is a synaptic pruning which happens. So that is pruning. Okay, that is synaptic pruning in adolescents, and which is malfunction. There is a malfunction. जिनकी होनी चाहिए उनकी नहीं होती, जिनकी नहीं होनी चाहिए उनकी हो जाती है, and that is what how schizophrenia takes place. That is why it is called as neurodevelopmental. In strict sense, neurodevelopmental we preserve it for first 5 years of life, but schizophrenia is an exception. The first decade of life.
So that means the synapses which should have been there for normal communication and progress are actually not getting developed. So that is how I related to the
The TAD concept, how it is important? How it is relevant?
So TAD concept, it is said that basically it is. So sir, there we have the CPCA. CPCA basically it prevents this TAD, this whole TAD to interact with the neighboring TADs. Now, if there is any changes here, so that would lead to some transcription
issues. Disturbances in schizophrenia. Seniors, your comments?
Neuroplastic period of time. So it has seen that in mothers who are deficient in folate and vitamin D4 can become deficient in blood donors. And the offspring of such mostly females are more into blood.
Good. Very good. Always there is a feedback regarding perinatal. I think perinatal psychiatry is
So this concept of then I have to study the genetical factors as well as the environmental factors, shared environmental factors, like not shared environmental factors. So you can have to differentiate the ecology with the genetical factors of the environment. That genetical factors because the psychologists will have more corresponding to more of the
how anything you know that will use. So one has done that kind of a therapy. So where in this gene silencing, genes, genetic therapies will come.
So genetic therapy.
Is there any scope?
Yes,
The
sir.
current understanding.
Yes, sir. I think that they can come up in methylation and deacetylation processes. Those enzymes which are responsible, if they can be inhibited. So
The understanding is too primitive to actually allow
yes, sir.
us. Because they are still not pinpointed.
Hmm.
Genetic therapies is silencing, silencing a gene, cutting the gene, replacing a gene by another gene. So we need to have that concrete, a very clear understanding. Where the pathology is still we are at conceptual stage, the evolving stage. So that is why, as for now, it doesn't have no role of any stem cell therapy, no role of any genetic therapy. Yes. Sarthak? You need to Srinivas.
Born in a slum area, he cannot when he has adult age, he feels that I cannot be able to get a money, so easily he can develop the schizophrenia during this stage. So due to the community's like a kind of poverty and other aspects of social problems also are developing schizophrenia.
So you will teach us. Maybe आप research the social theories के बारे में implications हैं. Is there you want to study this?
So there was an example when you quoted. There was a research which showed that migrants they are more predisposed of getting schizophrenia. For example, there was a study in London which showed that if the migrants' psychosis is removed, so the incidence of psychosis in London would fall by 32%.
Migrant patients they develop psychosis.
Yes, ma'am, they are more predisposed. That is the epigenetic symptom. That is because of
Task of schizophrenia patients who get migraine.
Senior migrants to couple of patients.
Because sleep is a predisposing factor.
So who sleep less, they are
Yes, ma'am. Sleep disturbances, diet disturbances, stress.
Very nice to meet you. It was very complex. मतलब एक बार में मुझे सब समझ में आया ये अच्छे से पढ़ना पड़ेगा तो genetics is something which is very complex and not something that we need to but very nice.
And that's nice presentation. I think that when this gene mapping was going on, it was just ending up on the sort of buzz that we will be able to find the cause of number of diseases, and then with gene therapy we will be able to even find cure for number of diseases. But this has not, that gene mapping has not resulted in that much of the regulation. It is because now we know that just the presence, merely the presence of hereditary absence of gene does not mean that this will be phenotype will also be there. So epigenetics, I think this is a new branch which is upcoming. Maybe we will be able to find the causes also and the intervention in the future. Definitely we should be aware of it.
So I think we agree with what Sir says, that said that genetic therapies have not worked because there is a huge role of neuroepigenetics or neuroepigenome, which we say. So that is why if we cut the gene, patch the gene, but the epigenetic modifications are not taken care of, still the person might land up even in more problems. So that is why this is very interesting area, evolving area. And we all know that brain, as you shared, 20,000 genes. So 6,000, 7,000 dedicated to CNS. CNS shares 6 to 7,000 more with other parts of the body. So around 14,000 genes are dedicated in a way to only CNS. So CNS, that is why it's very complex. We are not able to pinpoint the pathologies like in other disorders. Presentation wise, I think you have a very good acumen of teaching. So I think in that sense, when you present, we feel that we are being taught something. It's a very rare quality. So very good presentation. Adjusted to this is a very difficult topic. And I think genetics needs a different context of series of webinars or seminars in this time. Thank you.